So, From the Origin of Life to the Great Oxidation Event, episode 7. In this episode, we're going to look at what we can actually say of what happened during the Archean. How did the Earth transform from a totally anoxic planet, no oxygen, no animals, no plants, to a planet that had oxygen and started to be a good place for more complex forms of life? So we have to think, reflect a little bit of the types of approaches we have for dealing with the Archean. So we make use of two approaches. One is deduction. So that means testing hypotheses through experimental observation, right? If I hypothesize there were mammoths in the Archean, do I find fossils from mammoths in these very old deposits? Do I find molecular fossils, molecular phylogeny and isotopes? Those are the types of evidence we're going to look at for microbes. But then there's also induction, and that means that we try to generalize from existing observations. And this is, for example, uniformitarianism, so inferring the past based on observations of the present. And we've reflected a lot about the present in the first 3 episodes. So we'll see how those things work out. So let's go through our evidence. Can we say things about bacterial evolution and events looking at fossils? So do microbes leave fossils? And the answer is we don't know. We sometimes think we see fossils, but it is very debatable. So when you go to like microfossils, so this would mean looking through the microscope and looking for things that resemble our current cells. Very controversial. It has been shown that inorganic processes also can make things that resemble cells. So it's hard to say. Sometimes today's microbes make bigger structures like stromatolites. So these are kind of little islands of algae that grow, for example, in oceans. There's calcifications that make these structures. You see a picture of them here. And then when we go to fossilized stromatolites, now we're looking at bigger structures, more recent deposits or formations give reasonably credible specimens of fossilized stromatolites. But then when you go to the very old stromatolites, like 3.5 billion years old in the figure on the left, it becomes a little bit more controversial, but still more credible than the microfossils. So if stromatolites existed 3.5 billion years ago, does that mean that photosynthesis had already evolved then? Or were these other bacteria that made kind of similar structures? We don't know. Now, apart from looking at, let's say, shapes, we can also look at the chemistry. So do these organisms leave behind molecules, so chemofossils, in ancient rocks? And this could actually say something, because prokaryotes and eukaryotes and bacteria and archaea, they consist of different molecules. For example, that membrane that surrounds a cell in bacteria and eukaryotes, those membranes are made of esters, of fatty acids. But in archaea, they're made of isoprenoid ethers. So if we could find that type of molecule in ancient rocks, we could say something about whether archaea or bacteria or eukaryotes had already evolved at a certain moment in time. So a lot of people have tried this. Turns out diagenesis and contamination cause problems. Also, it turns out biomarkers that are supposed to be specific for a certain group of organisms often turn out to be less unique than believed. So if you find an ether, it doesn't actually mean it belonged to an archaea. It could also have been present in a bacterium because some bacteria have these lipids. And then the more informative molecules, so DNA and protein, do not preserve that long. So if you would go to a mammoth that is 10,000 years old, you can still extract its DNA if it's preserved well. That's pretty good, but that's about where it ends. When you go to proteomics, or the proteins, you can go back even millions of years. So when you have a well-preserved dinosaur fossil, you can sometimes determine protein sequences. And this study, for example, shows the relationship between dinosaurs and birds based on protein sequences. But that's where it ends. It doesn't go back more. So unfortunately, we cannot characterize DNA directly from 2.5 billion years ago. But as discussed in episode 3, we can use molecular phylogeny. So this is building evolutionary trees. And so let's look at that origin of eukaryotes again. So this tree on the right, made by Laura Huck, shows eukaryotes as originating from archaea. So what that would mean is that eukarya evolved later than bacteria and archaea. Now, this is still very controversial. Not all investigators yet believe this, want to believe this, but the evidence becomes pretty strong. So in 2005, investigators identified a group of archaea in marine sediments that have never been found before. They're only known by their DNA sequences, but they clearly have genes that more closely resemble eukaryotes than any other prokaryote ever found before. So when you build trees, you now see that the eukarya originate from this group of archaea. So most likely, bacteria and archaea evolved first. And then at some point, eukarya evolved from archaea. Much later, what happened is that these eukarya that can engulf other organisms, one of these organisms enslaved a alpha proteobacterium similar to Pelagibacter and started using that alpha proteobacterium to respire. So now this eukaryote became endowed with respiration. Couldn't do that before. And then again later, one of these eukaryotes could respire, did the same thing again. Now it took the cyanobacterium captive and converted it into a chloroplast. And so that endowed that eukaryote with photosynthesis, giving rise ultimately to plants. So we can actually say something about what happened. The eukarya happened very long ago. I don't think anybody yet inferred when that might have happened. But they did that for photosynthesis. So the question is, when the Great Oxidation Event occurred, was that because cyanobacteria evolved and photosynthesis started on Earth? Or did cyanobacteria evolve much earlier? Just the oxygen never accumulated in the atmosphere. And that is a question that has been, I think, quite convincingly addressed with molecular phylogeny. So what these authors did is they took the phylogeny of the bacteria and the cyanobacteria and the plants, and so what became clear is that cyanobacteria has a sister group of bacteria that are incapable of photosynthesis. So cyanobacteria evolved from an ancestor that couldn't respire or couldn't make oxygen. Now, when did this happen? And these authors kind of anchored that phylogenetic tree by measuring the rate of evolution for the plants. And what this shows is that the current cyanobacteria we know came about after the Great Oxidation Event. But the separation between cyanobacteria and their non-photosynthetic ancestors happened before the Great Oxidation event. So this study suggests that actually, as soon as the cyanobacteria evolved, the oxygen, at least not much later, started to appear in the atmosphere. So I think that is a very powerful step forward. Now, another really important way and powerful way to actually say things is using isotopic fingerprints. So, and the isotopic fingerprints that are shown in this bottom graph here relate to sulfur and carbon. What the top graph shows is that is in red, kind of the old picture, and in blue, the more modern picture. And the main difference between those two, I would say, is that the oxidation event happened more gradually than previously assumed, but also that there might have been some spikes of oxygen before the Great Oxidation event. What the isotope data show is that up until the Great Oxidation event, the isotopes of sulfur, so it's mainly related to sulfur 33, are only consistent with a oxygen-less atmosphere. So these deposits could only have been built up without any oxygen in the environment. Otherwise, you would never get this pattern of 33 sulfur in your deposits. Okay. Now the other interesting one is carbon. So when life fixes carbon, so this enzyme Rubisco, we talked about it before, the Rubisco likes or prefers or has a higher rate for the 12 carbon. It has a lower rate for the 13 carbon. So what happens when a plant or a bacterium fixes carbon is that it becomes a little bit depleted in 13C, and the remainder, so those would be the carbonates you find in rocks, becomes enriched in 13C. That is what Rubisco leaves behind. So what you see after the Great Oxidation event in the sediment record is that suddenly you see a large accumulation of 13C, which can only be explained, or which is very consistent, let's put it that way, with a very strong increase in biological carbon fixation. So I think the isotopes very strongly constrain the what happened, let's say the Great Oxidation event in time. It must have happened around that time. Okay. So constructing the Archean depends on both deduction and induction. Fossils, microfossils, and chemofossils are debatable sources of information about the Archean, but sophisticated phylogenetic analysis indicate that evolution of oxygenic photosynthesis may have directly led to the Great Oxidation events. Similar analysis indicate that eukaryotes evolved from Archaea, so came about later, and the sulfur isotope signature of Archean rocks show near complete absence of oxygen until 2.5 billion years ago. Enrichment of 13C in carbonates provides evidence for high rates of CO2 fixation since the Great Oxidation events. All right, that concludes episode 7.